Using humanized mouse models to study cleft palate, biological and ethical considerations. This video was created by Juliet King and Scott Williams' lab at the University of North Carolina at Chapel Hill. Welcoming a new child into a family can be exciting, but many parents worry if their child will be born with a birth defect, a disease that arises during the development of the baby. Did you know that cleft lip, with or without cleft palate, is one of the most common birth defects seen in newborns, both in the US and globally? Cleft lip and cleft palate occur when the oral and nasal cavities fail to separate during development. This process is known as palatogenesis, or the development of the palatal shelf. Individuals affected by cleft lip and palate experience high rates of infection, require multiple surgeries, and necessitate years of speech and emotional therapy as a result of the disease. Thankfully, many countries are adopting the model to establish cleft lip and palate care centers that specialize in treatment, providing continual care at the local level. These approaches improve access to care and alleviate the dependence of developing countries on the resources of wealthy countries. One reason we see so many cases is due to the wide breadth of genetic mutations that disrupt the process of palate closure. Run your tongue around in your mouth. What you're feeling is your oral epithelia. You might not expect it, but genetic mutations that affect this part of your mouth have recently been appreciated to cause cleft palate. As a developmental biologist, I study the genetic changes in individuals affected by cleft palate and determine how these alterations impact the proteins and cells within that person's body during development. One ethical approach to studying palatogenesis in humans is to generate humanized mouse models. Instead of having to remove tissue from an affected individual and culturing those cells in a dish, we can understand how specific disease mutations affect mouse palatogenesis in the context of an intact tissue. Our lab has developed multiple novel models of cleft palate in mice using viruses as a tool to alter gene expression in developing mouse embryos, a tool we call luggage. This approach allows us to express human disease proteins in all outer epithelia, like the oral epithelia as well as the skin. The way we do this is to generate small pieces of DNA that encode the human protein we're interested in studying. Then, with the help of the virus, we can stably incorporate the DNA into the host oral epithelia through an in utero injection. This ultrasound video demonstrates how we introduce the human cDNA through an injection into the developing mouse. Supporting research efforts designed to untangle the genetic underpinnings of this complex developmental disease could open the door to novel therapies or improved early detection and genetic screening methods. To conclude, technology like the one developed in part by our lab pushed cleft palate research in new, more ethical ways, thereby improving the well-being of affected individuals and the animals we use to study palatogenesis.